Hi, my name is Bailey Dorman, and today we're going to talk about the bacteria Clostridium botulinum and the disease it causes called botulism. What is Clostridium botulinum? Clostridium botulinum are bacteria that are gram-positive, rod-shaped, and spore-forming bacteria that grow in low-acidic, low-oxygenated environments such as soil. They release one of the seven deadly neurotoxins classified in the letters A through G, and their endospores can be found in contaminated wounds, contaminated food, and in the GI tracts if ingested. The endospores in this bacteria release one of the most deadliest toxins known to man. 30 grams of this potent toxin could actually kill every person in the United States. The toxin is classified in letters A through G because there are 7 different unique deadly toxins, but only A, B, E, and F toxins can cause illness in humans. Once the toxin is released, it travels down to the muscle cells and attacks the receptor sites there, preventing a muscle contraction from happening, ultimately causing paralysis. In the top left corner, you see a nerve cell attaching to a muscle cell, and in the top right corner, you will see a closer image of this neuromuscular junction. In the nerve cell, there are tiny little sacs called synaptic vesicles that contain the neurotransmitter acetylcholine. Once this acetylcholine is released, it travels across the synaptic cleft, which is that space you see right there, and attaches to the receptor sites in the muscle cell. Ultimately, this causes a muscle contraction. But when the botulinum toxin is released into the body, it binds to these receptor sites, preventing acetylcholine from being released. Ultimately, this causes paralysis or flaccid paralysis, and muscle contractions cannot happen. Once it binds to the receptor sites, it irreversibly binds, and there's no going back from that. The endospores in the bacteria Clostridium botulinum can release their deadly toxin and intoxicate our bodies in different ways. They can release their toxin that then intoxicates the food that we then ingest, then causes the toxin to spread throughout our bodies, or we can ingest the endospores through the food we eat that then colonize in our body and release its toxin. Therefore, this results in 3 different kinds of botulism: foodborne botulism, infant botulism, and wound botulism. Foodborne botulism is caused when the endospores of the bacteria are not destroyed during food processing. The endospores can contaminate foods such as improperly canned foods, meats, and fish. When the endospores are not killed, they release the deadly toxin that is virtually colorless, odorless, and tasteless, and when ingested can cause symptoms such as dizziness, weakness, blurry vision, abdominal pain, constipation, followed by progressive paralysis. Approximately 25 foodborne botulism cases take place in the United States each year, and nowadays are caused by unrefrigerated homemade salsa, garlic and oil, potatoes wrapped in aluminum foil, and improperly prepared fish. Infant botulism is caused when the endospores of the bacteria are directly ingested into the body. Once inside the body, they travel down to the gastrointestinal tract and colonize because infants have yet to develop the proper bacteria to fight them off. When the endospores colonize, they release the toxin that then causes symptoms such as crying, lethargia, constipation, and then eventually muscle weakness. Some of these symptoms are often confused with other infant-related illnesses and are not treated in time, which then result in the death of the infant. Most infant deaths are often confused with sudden infant death syndrome, and approximately 100 cases of infant-related botulism occur in the United States each year. They most often are linked to the ingestion of honey, so parents are advised not to feed their infants honey if they're under the age of 1. Wound botulism takes place when the endospores of the bacteria contaminate an open or improperly cared for wound. When endospores contaminate this wound, they release their toxin, which then symptoms usually follow 4 more days after contamination. The symptoms are much like the physiological effects that foodborne botulism has on the body, which include dizziness, weakness, blurry vision, abdominal pain, constipation, much of the same. Approximately 30 cases of wound botulism take place in the United States each year, most of which are linked to the black tar heroin epidemic that takes place in California. The botulism disease can be diagnosed in two different ways. One is in a culture sample, and the other is in a mouse bioassay. In a culture sample, a sample is obtained from a contaminated source of food, feces, or wounds, and then brought into the lab to confirm botulism. In the other test, you have a mouse bioassay. A mouse bioassay is when a sample is brought into the lab and divided into two parts. Sample 1 is then injected with an antitoxin, and sample 2 is left contaminated. Then you bring in two mice. Mouse A is then introduced to the antitoxin sample 1, and mouse B is introduced to the contaminated sample 2. If mouse A lives while the other one dies, then botulism can be confirmed. If botulism is confirmed, it must be treated quickly and aggressively in order to prevent paralysis or death. Treatment methods include washing of the gastrointestinal tract or pumping of the stomach in order to get rid of all the endospores that have colonized there. The other is passive immunotherapy, which is the administration of the antitoxin into the blood, which then neutralizes the botulism toxin, preventing further nerve or muscle damage. The other is the administration of antimicrobial drugs that then kill the bacteria Clostridia in the GI tract or in the wounds. And the other is just the basic proper care of an open wound that you should, do anyways. So it keeps the endospores from contaminating the wounds. Botulism is caused when endospores of the bacteria are not killed during food preparation or food processing. Ways to prevent botulism is by heating food to its decimal reduction time that kills the endospores, refrigerating food to prevent the germination of endospores, establishing an acidic environment for which the endospores cannot thrive, or following basic USDA regulations for proper food care and processing. The Clostridium botulinum bacteria and its botulism toxin has posed many benefits to the cosmetic industry of the medical field. The botulism type A toxin is brought into the lab and gone through an extensive purification process in order to produce a non-lethal prescription drug. This drug is administered in extremely small doses into the facial muscles. Once administered, it eliminates wrinkles and causes smooth skin in a process called Botox. Once Botox treatments have started, they must be repeated every 6 months in order to maintain the desired effects. In conclusion, Clostridium botulinum is a spore-forming, anaerobic, gram-positive bacteria that produces 7 unique deadly toxins, all of which are physiologically undetectable. The endospores formed by this bacteria can manifest themselves and contaminate food, wounds, and GI tracts. Once the endospores release the deadly botulism toxin, it must be treated quickly and aggressively in order to prevent lethal effects. And although the botulism toxin is one of the most deadliest toxins scientifically proven, it is also one of the most widely used medical treatments in the cosmetic industry by men and women all over the world. So, this is the bacteria Clostridium botulinum and its bacterial disease, botulism.